Our next presentation is going to be from a team of investigators also from UCSF. And they're comprised of Martin Kampmann, David Kokel, and Michael Keiser. And as in years past, you'll be hearing from all of them for their presentation.
Thank you, Kathy, and the whole team for putting together this exciting symposium. Obviously, we are also very sad and grateful for Paul's support that I think has made a big difference for all three of us in our research programs, and we'll tell you about that now. We are also from the neurodegenerative disease cohort, and we decided to actually focus on the protein that is one of the two hallmarks of Alzheimer's disease, tau, together with A-beta, but it has been much less investigated as a potential therapeutic target. Recent data suggests that a potential mechanisms driving disease progression is the aggregation and almost prion-like spreading of tau aggregation from cell to cell. And the three of us combined our different technologies and our different backgrounds to try to define cellular mechanisms and also potential therapeutic strategies around the cell biology. And I'll start off by telling you about our genome-wide screening approach based on CRISPR-I and CRISPR-A and genetic interactions. And then Mike will follow on and tell you about his systems pharmacology approach, and then Dave will tell you about zebrafish screening. As a postdoc in Jonathan Weissman's lab, I co-developed a new way to screen mammalian cells in a very specific manner for loss-of-function screens that we called CRISPR-I. We're knocking down endogenous genes using a catalytically dead Cas9. We can also do gain-of-function screening that we called CRISPR-A. And our goal for this project was to really fill in the question marks along the cell biology of why does tau aggregate in cells? How do tau aggregates spread across cell-cell boundaries to seed more tau aggregation? What are clearance mechanisms? And finally, a big important question, obviously, why are these tau aggregates or some species along this process toxic to neurons? And you can think of each question mark as a molecular mechanism and cellular mechanism that would teach us some interesting cell biology. Some of the genetic modifiers that are now being identified at an accelerating rate for Alzheimer's disease might act along this pathway. Importantly, Amy already introduced the concept of selective vulnerability, which to me is one of the most exciting questions. Why is it that whereas even in a patient, for example, with a familial mutation in tau, those tau mutant proteins are expressed throughout the brains, yes, they are very specific cell types of neurons that are specifically sensitive. I think if we could understand that puzzle, we could understand also how other neurons are maybe resilient and how we could use that knowledge for therapeutic benefit. So again, some of these mechanisms of cell type selective vulnerabilities might be some of these question marks, and so ultimately each of them is a potential therapeutic target as well. In previous talks, I told you about how we built HEC293 cell models, and were actually successful to identify several steps along the pathway. We were able to identify the cell surface receptor for tau uptake into other cells, which happened to be a very specific lysulfated heparan sulfate proteoglycan. In unpublished work, we also defined escape from the endolysosomal system as a rate-limiting step in this prion-like spreading. But what I really wanted to emphasize today is how the support and input from the Allen Foundation pushed us to say, we want to not look at this in HEC293 cells. We want to look at these processes in human neurons. And so based on the support from the Allen Foundation, we were able to establish our CRISPR-I, CRISPR-A screening platform in iPSC-derived human neurons to look really at these things in the relevant cell type and to be able to get at questions of selective vulnerability. This is our first pilot screen that we did in the system. We generated stable iPSC lines expressing CRISPR-I machinery. We can transduce them with a guide RNA library. And then in this first experiment, we were simply looking at survival. And we had two branches. We passaged the iPSCs to ask what genes are relevant for iPSC survival, and we took at different time points samples from neurons of different ages. And by quantifying using next-generation sequencing the frequencies of guide RNAs observed in these populations, we could detect genes that, when targeted, would lead cells to drop out of the population at higher rates. And again, I don't have much time to tell you a lot of the details. We hope to publish this very soon, actually. But the first question we wanted to answer is, are there separate genes that control neuronal survival from, say, iPSC survival? And that's certainly the case. While there are some globally essential housekeeping genes, we found neuron-essential genes. If we ask what types of genes are enriched in those neuron-essential genes, actually from an unbiased pathway enrichment analysis, 3 out of the top 5 genes, gene sets were associated with neurodegenerative diseases. So we do think we can get at specific biology here. We wanted to more deeply understand what these genes do. And so we built 2 platforms along that. One of them couples CRISPR-I screening, where we perturb genes with CRISPR-I to single-cell RNA sequencing, so that for each gene that is a column here that we knock down, we can monitor differentially expressed genes. So each column here is a different gene in the transcriptome that we capture by single-cell RNA sequencing. Again, I don't have much time to go into this. We can do hierarchical clustering that reveals both modules of genes that we perturb and modules of the downstream regulated genes. We can see that there are actually very interesting differences between iPSC and neurons. And the second platform, together with Michael Ward at the NIH, was that we did high-content imaging of neurons in which we expressed either non-targeting guide RNA, or in this case, a guide RNA that is against a pretty globally essential gene in the electron transport chain. And so we can track survival over time, even of individual cells, along with morphology. Can do shawl analysis and other things to more deeply characterize these genes. Interestingly, we found one gene in our screen that actually improved neuronal survival very significantly. This is just a bird's eye view, wild type versus knockdown of MAP3K12, also a kinase that's neuron selectively expressed, also called DLK. And that was known previously to improve neuronal survival, but it's nice that we rediscovered it in our screen. And now we can take patient-derived cells that have familial mutations, make CRISPR-corrected isogenic lines, differentiate into them cell types, and do modifier screens for some of the cellular defects that are linked to the disease mutations. And last slide, Avi Samuelson, a postdoc in the lab, showed that, for example, in tau mutant cells that have a V337M familial mutation, forms of tau, such as phosphotau or tau oligomers that are found in patient brains, including Alzheimer's patient brains, accumulate selectively. And he established a fax-based protocol, so we can now do modifier screens that ask what are cellular pathways in those neurons that would either promote or prevent accumulation of these disease-associated forms of tau, using antibodies that are the same antibodies that pathologists use to diagnose the disease. And using a small pilot library for chaperones and co-chaperones, he already found things that, for example, strongly repress formation of these tau oligomers.
All right, so picking up the second part of our 3-part talk, we're coming from very different perspectives in trying to get at mechanisms of these neurodegenerative diseases. And the perspective I'm going to talk about is molecules. And so what we started with was a collaboration with Stanley Prusiner at the IND. And in this collaboration, what they had already done, along with Daiichi Sankyo, a pharma company, is screened a rather large collection of molecules, novel molecules, for the ability to, starting with one clone, when exposed to a lysate from a second clone that contains overexpressed YFP tag tau, look for molecules that would inhibit this process. And so this worked, and out of this screen, various campaigns of medicinal chemistry have emerged, but there's a really big problem that we need to deal with in this if we want to use all of the information from this screen or discover something new. We know from the screen that we can go from compound to phenotype, but not just for this, but for any phenotypic screen like this, how do you go backwards from phenotype to compound? How do you find mechanism? How do you optimize on this mechanism in a knowledge or information-guided way? And so one way to do this, and what the focus of our sub-project had been, is to figure out the protein targets the cellular machinery targets that are intermediate to this process. Some molecules interact directly with tau, or with alpha-synuclein at another screen. Many do not. And so what we were able to do is use various statistical methods, which, again, due to time, I'd love to talk about afterwards and separately, to relate small molecules to protein profiles on the one hand, and then to relate protein profiles to phenotypes. And these are these enrichment factor guilt by association metrics that we originally developed for side effect prediction in previous work. And so Garrett Gaskins was someone who adapted this to a cellular screening context. And so we did this on a preliminary tau screen. And indeed, the networks that we found this way, in a very unbiased way, straight from the screen itself, actually tied into lysosomal function, as Amy mentioned earlier. We had examples that seemed initially strange, like trip channels coming up, which doesn't make that much sense from the perspective we were coming from. But it turns out that there are certain trip channels, like TRPM, mucolipin, that are expressed on the lysosome. And so the molecules and the computations kept leading us to these networks. But there was a real problem at this level, which was toxicity, because we hadn't explicitly been trying to correct for that. However, in the screen, we have this data. And so if you imagine what we're doing, we had this axis of phenotype from molecules that, in an undesired way, actually promote various cellular proteinopathies, all the way through those that we're looking for that block it. And in this space, if you look for enrichment off to the right towards that purple star, that's the way we had gotten these networks. And by the way, I should mention that molecules that we purchased to phenocopy and test worked, but again, had toxicities. But really, if you knew more of what's going on, many of the compounds are achieving these effects through mechanisms that are affecting cellular viability, which is perhaps no surprise. This propagation will go down, the fluorescence channel will go down. So what if we opened it up? In the case of these screens, we have counter-screening data for toxicity. This defines a phenotypic space. The y-axis is toxicity at the top versus actually molecules that promote cell growth at the bottom in that counter screen. And the x-axis is exactly as shown above. And in this space, where every dot is a compound from the screen, and we have hundreds of thousands of compounds that have got us to this point, and then on the order of 5,000 or so compounds that make it to this point in the screen, what we could do is we could ask, are certain regions of this phenotype enriched for proteins that we can go and test? And so if you look at this, it turns out that they are. And this is an example where a heat map is constructed by looking across that whole space, calculating the enrichments, and then going and doing experiments. And so one example from before, mTORC1, that should be familiar to many of us here, is enriched as a target that underlies specifically molecules that inhibit proteinopathy, but exclusively in the region of toxicity. And indeed, this is a very toxic target. So what we became interested to do then was to look for things more like the plot at the right, which is if we run these calculations and generate these plots and do all this work on every protein that we have enough ligand information for independently, some of them will have enrichment patterns where regions of the phenotype space where molecules are not toxic or even promote growth and yet are also enriched for the activity we want will be there. And so we had an example of this. This is very hot off the presses just a couple weeks ago, so I can't yet say what target X is. I'd love to follow up with those of you who are curious afterwards once we're able to, but this is very recent, where what we were able to do was find protein targets that we wouldn't have thought to look for, and then go buy molecules by phenocopy experiment that are known for this target as a starting place right there, buy it right off the shelf, put it into the screen. And so we did 3 of these phenocopy experiments for this, and all 3 of them worked, which, as a computer scientist, was shocking. I did not expect all 3 of them to work. I hoped one might, sort of. They all worked. They were all sub-micromolar, often even below 250 nanomolar, and they exhibited no toxicity whatsoever. And so this was a big change for us. And I should mention this is in the alpha-synuclein screen because there's multiple screens that are going on, and we decided to take these techniques and apply them across the board. This was a big change from the first tau screen analysis where the molecules had worked but had been toxic. And so I'm very interested in this angle of multiple phenotypes and then bringing these systems pharmacology methods to bear. We've used statistical methods for this so we could interpret it. One direction that we're going now based on this foundation is machine learning approaches to go for both of these steps. And then finally, before I turn it over to Dave, what we see from this is an engine, a pipeline that we think can apply not to any particular screen, but as a general approach for therapeutic discovery, where we can move from a cellular screen or a whole organism screen, as we'll hear about soon, look at molecular structure. And the majority of the work I've shown you was 2D, but along the way, one of the things I didn't talk about was developing a 3-dimensional representation of small molecules for machine learning, deep learning, and statistics. From there, for instance, using the statistical SEA method, predict the target profiles, look at these enrichments, and then loop back in this virtuous cycle, where we can subject every prediction to direct experiment, even in an off-the-shelf way. And then as we do that, there's no reason we have to restrict ourselves to known molecules. There's already 500 million molecules that you can buy off the shelf. And beyond that, this can be a synthesis guiding engine. And so from there, we're very interested in moving this into new therapeutics. But every step of the way, one thing I've learned to appreciate as a computer scientist is needs to be tied to experiment. So thank you, and here's Dave.
So Mike and Martin are both finding lots of genes and compounds that we'd like to test in living animals. So I study zebrafish and behavior-based neuroactive drug discovery. Fish are small and they easily absorb compounds, so they're really great for large-scale chemical screens. They're vertebrate animals like us with complex brains and behavior, and this is an example of what a behavioral assay would look like in the fish. This is a 96-well plate, fully loaded with zebrafish with 8 animals per well. And when I play the movie, you'll see how these animals respond to the stimuli. It'll be indicated on the bottom. So the light will turn on. There's a strong behavioral response. The light turns off, and the animal calms down. We can extract patterns of motor activity from each well. And we don't just look at one behavioral assay, but a whole battery of behavioral assays to try to get a high-resolution profile for how different compounds or genes affect zebrafish behavior. We're trying to get fingerprints for specific pathways and compounds. So here, the hypothesis was that we could overexpress human disease genes in the zebrafish and then find some biochemical, cellular, and behavioral phenotypes in these animals and then do some chemical screens to try to find compounds that suppress neurodegeneration. We overexpressed the mutant tau lines, and they localized to the right place. We could see tau localizing to the axons, more so than just the GFP controls, which were primarily in the neuron cell bodies. We saw increased phosphorylation in the neurodegeneration models compared to the controls. But when we looked for cell death, we didn't see a great increase in the amount of cell death in the neurodegeneration models. So we started to look at different types of environmental factors, different compounds we could add to the fish that would either exacerbate the cell death phenotypes or block cell death. And one of the types of compounds that we started to look at were NMDA receptor antagonists. These compounds block excitotoxicity. They're neuroprotective. You can see here in the control animals, these fish, they don't respond very much to this repeating acoustic stimulus. When I play the movie, you'll see they'll respond to the first couple stimuli, but then really not to any of the others. By contrast, if we treat these animals with NMDA antagonists and then run that same set of repeating acoustic stimuli, you can see that these animals are going to respond to every single stimulus in the battery. It's a really striking behavioral phenotype. And so we wondered if we could use this for a larger scale screen. We did a pilot screen of 700 bioactive compounds in different neurotransmitter signaling pathways, and 4 out of the 5 top hits were NMDA antagonists. So we wondered if we could increase the screen to novel compounds and find novel compounds that cause this kind of phenotype. So now we screened 10,000 novel compounds, and we have 50 novel hit compounds. I'll show you a couple examples from two classes. The first class of hit compounds, you can see compared to the negative controls, which respond to low frequency stimuli, but then as the stimuli become higher and higher frequency, they stop responding. We have this class of hit compounds, which allow the animals to keep responding at this frequency. Also, we found a class of hit compounds. We found only 2 of these compounds out of the whole 10,000 screen where the animals are now responding to the highest frequency stimulus that we give the fish. So now we're trying to figure out with Mike and others how these compounds are working and to see if they are neuroprotective. So overall, the 3 of us are working together to try to integrate these technologies in systems pharmacology, functional genomics, and large-scale phenotyping. Thanks.